Today, we're going to examine a key discovery in the field of amyotrophic lateral sclerosis, or ALS. We're drawing from a recent study in Nature Neuroscience that uncovers a fundamental difference in how the body's cells respond to the two main forms of this disease, and this difference could really change how we think about developing treatments.
So here's our plan. First, we'll quickly review the puzzle of ALS itself. Then we'll zoom in on a specific cell type, the microglia. After that, we'll explore the study's main finding about two very different cellular responses. We'll look at the mechanism behind that difference, and finally, we'll discuss what all this could mean for future treatments. All right, so to really appreciate what's new here, we first need to lay a little groundwork on what ALS is.
At its core, ALS is a neurodegenerative condition. What that means is it's defined by the progressive loss of motor neurons. Now, these are the nerve cells that control your voluntary muscle movements. As these crucial neurons in the brain and spinal cord degrade, the muscles they're connected to start to weaken and waste away.
Now, here's a really critical point. Although the symptoms often look similar, ALS isn't just one disease. On one hand, you have sporadic ALS, or SALS. It's the most common form, and it seems to appear without a known genetic reason. On the other hand, you have C9 ALS, which is the most common genetic form of the disease. It's caused by a very specific mutation in a gene called C9orf72. This distinction is really the whole basis for the findings we're about to get into.
So, to understand what makes these two forms of ALS so different, the researchers decided to focus their attention on one particular type of cell.
And those cells are the microglia. You can think of them as the resident immune cells of the brain and spinal cord. They're sort of the dedicated guardians and cleanup crew. Their job is to respond to any injury or disease by starting up an inflammatory response and clearing out cellular debris. In diseases like ALS, figuring out exactly what they're doing or not doing is a huge area of research.
And here's the crucial link. The study confirmed that the C9orf72 gene, the one that's mutated in the genetic form of ALS, is most active, or what scientists call highly expressed, right inside these microglial cells. This finding gave them the perfect reason to focus squarely on the microglia to see how that mutation might be driving the disease.
And this brings us right to the heart of the discovery. When the researchers compared the microglia from patients with sporadic ALS to those with C9 ALS, they saw a really stark difference in how these immune cells were behaving.
And the contrast is quite clear. In sporadic ALS, the microglia seem to be doing their job. They mount an active defense, shifting into a reactive state to fight the disease. But in C9 ALS, that response is significantly weaker. The microglia just don't seem to get the message to activate, and they mostly stay in a more passive, resting state.
You can see this difference visualized here. The data clearly shows a much higher level of microglial activation in sporadic ALS compared to the genetic form. It's as if in one disease the immune system is actively fighting back, while in the other, it's been hobbled.
This quote from the paper really sums it up nicely. While the microglia in sporadic ALS are able to switch over to a responsive, disease-fighting mode, the microglia in C9 ALS are far less capable of making that change. They're essentially failing to answer the call to action.
So the next logical question is, why? What's going on at the cellular level that stops the microglia in C9 ALS from activating the way they should? Well, the study offers a pretty compelling explanation.
The mechanism seems to be something called haploinsufficiency. Now, that sounds complex, but the idea is actually pretty straightforward. We all have two copies of most of our genes. In C9 ALS, that big genetic mutation basically knocks out one copy of the C9orf72 gene. This creates a loss-of-function state, meaning the cell just can't make enough of the C9orf72 protein to do its job properly.
And this shortage of protein has a very direct effect. Researchers saw that this loss of function specifically messes with what are called the endolysosomal pathways inside the C9 ALS microglia. And this is a really critical piece of the cell's machinery.
You can think of this pathway as the cell's waste disposal and recycling system. The first step is that the microglia swallow up material, things like damaged proteins or other cellular junk. Then, that material gets transported to a part of the cell called the lysosome, which is like the cell's recycling center. Finally, enzymes inside that lysosome break the material down.
What the research suggests is that in C9 ALS microglia, it's this final step, the breakdown of waste, that's broken. The cell is still able to take in the trash, but it can't process it properly. This creates a sort of functional bottleneck, where unprocessed material just builds up inside the cell. and that pileup might be what's preventing it from mounting a proper immune response.
So we've looked at the cellular mechanics, but what does all of this actually mean for patients? Well, understanding this difference is more than just an academic exercise. It has some pretty profound implications for how we might treat ALS down the road.
And what's interesting is that this sluggish immune response isn't just limited to the microglia. The study also found that astrocytes, which are another kind of important support cell in the brain, also show a similar blunted reaction in C9 ALS. This suggests we're looking at a much more widespread failure of the brain's support system in this form of the disease.
And this all leads to the central conclusion. We may be looking at two conditions that appear similar on the outside, but are being driven by very different problems on the inside. In sporadic ALS, the immune response might even be overactive, but in C9 ALS, it seems to be fundamentally broken. This strongly suggests that a single, one-size-fits-all treatment probably isn't going to work for everyone.
And so this leaves us with a final thought-provoking question. If C9 ALS is defined by a loss of proper immune function, maybe the goal for future therapies shouldn't be to suppress inflammation, but instead to restore it. The challenge then becomes figuring out how to repair that broken waste disposal machinery in the microglia and astrocytes to let them get back to doing their critical job of defending the brain.